This recorded session is dedicated to the completion lecture for our module on gene regulation. Um, as a review, we've already learned how important gene expression regulation is. We've talked about the rationale behind cellular organisms needing or processing regulation or manifesting regulation as a daily activity, as a daily process. At the level of course, gene expression, the level of producing proteins, of byproduct biomolecules. And also, we've appreciated that especially for the case of multicellular organisms, ma, the process is very evident. Okay, for humans, for example, we can point out that our somatic cells possess the same genomes. No, all our somatic cells will essentially possess the same set of genes, but it is gene regulation or gene expression regulation that is responsible for the profile, for the biochemical profile or the metabolic profile of different cell types. No? That a certain gene can be well expressed by specific cells, while a particular gene may not be expressed by those cells, and instead in another type. Okay, it is more expressed, or it's more active. No? Um, we pondered on the central question of how do eukaryotic cells decide which genes to turn on and off, primarily diving into the basic rationale, no, about signal transduction. We've talked about a little overview on this. Actually, for this week, we are going to have our asynchronous module on signal transduction. So you'll come to appreciate more of the process once you complete the module also for this week. No? Then, in general, we looked at different stages, or I would say levels or mechanisms where that eukaryotes use in order to regulate the genes that they possess, not to regulate the expression of genes in response to an input, in response to a signal. Okay, we have given some basic examples, like at the level of primarily the level of no no at the level of uh, gene expression. We have the factor of time. Okay, we've come to realize that cells can control expression of their genes acutely or in long term, depending on the context. No, now for mostly for established systems like cells or ano na no mga living tissues, we expect short-term control of gene expression, while for developing organisms in the context of let's say a developing animal, no, there's a no idea of long-term control. Also in the context of a maturing or aging animal or aging multicellular organism, the idea of cellular differentiation or tissue cellular differentiation connects very well with gene regulation. No? Okay, so this is just an infographic discussing or giving us. I've discussed this no last synchronous session last year. Then we identified the different elements, the differences between prokaryotic and eukaryotic gene expression. No, now I'm pretty sure this will come as a sort of as a review for everyone na no kasi we've already um, we've already covered a lot or you guys have had introductory I don't know introductory classes in genetics, So dapat familiar tayo with the prokaryotic gene expression regulation, yung concept of operon and also the major elements, no? In prokaryotes, as a review, we have these four important, I don't know, high-level ideas or high-level concept, high-level terminologies that we always go back whenever we encounter a prokaryotic gene, an example of a prokaryotic gene being regulated. No? So the concept of the regulator, the promoter, the operator, and the structural genes. So these are very important ideas or very important concepts that you have to have a basic grasp on. So I don't know, it will be a lot easier for you to understand specific examples. Now, I remember that I did not discuss in detail these two operons, the tryptophan operon and the lactose operon, because I put it in your hands or I delegate to you the responsibility of doing further readings about them. And I'm pretty sure you have also encountered these operons already in your genetics classes. Um, for this completion lecture, we will dive deep into the different mechanisms or levels of eukaryotic gene expression. And I guess, or if my memory serves me correctly, we have already covered the first one, no? The level of transcription. So we're going to cover the remaining five levels or steps that the eukaryotic organisms can actually intervene, or can actually not intervene but can how do you call this, steps or pathways that eukaryotic organisms can utilize in order to control how certain genes will be turned on and off at specific time points. So we've covered transcriptional control. um, we've reviewed elements such as promoters for both eukaryotes and prokaryotes, enhancers, and also for this two important elements, we've introduced the idea of combinatorial gene regulation, no? Where fine level or fine and coarse level control of gene expression is actually achieved by multiple interactions of promoters and enhancers for specific sets of structural genes. You know. As an example, we have talked about the galactose group of genes in the yeast, no? And I won't go over this topic anymore, because we already covered them last time. Okay. We just demonstrated or illustrated how promoters and regulators can or how specific genetic elements serving as promoters or enhancers can influence the activity of a gene set, no, or a structural gene in response to the environment, no, or in considering a proper response to a stimuli needs to be mounted or produced by the cell, by the dynamic system. And then I covered this already last time. Okay, I think we can already start with our with our second level, which is on RNA processing. No? So we can control, or there is control of gene expression at the RNA level, at the RNA processing level. So we will remember from our previous, or we can remember from our previous discussion, especially when we talked about the central dogma, how RNA is produced, no? And there are specific processes that are happening after transcription. We call post-transcriptional modifications that make our RNA transcript mature, or allow it to mature in order for it to, of course, be ready for translation. There are two independent regulatory mechanisms that happen in eukaryotic systems when it comes to RNA processing. No? So we have our polyadenylation, our alternative polyadenylation, or our alternative splicing. As a review, let's look into or again define these two events. Polyadenylation is where multiple A residues or nucleotides are added at the tail portion of the mRNA of the ano no, the messenger RNA molecule. Okay, so this poly A tail serves as a protective tail of some sort, so that exonuclease attacking at the 3 prime end will not really be capable of will not really be capable of attacking that portion of the mRNA. And then we also have alternative splicing. So you're, I don't know, just a review. Ano yung alternative splicing? Right? Remember that for eukaryotes, we have our express regions and our in-between regions that are not really expressed, that are not really contributing to the genetic code for peptide translation. So we have our exons that are spliced and put together in order to to come up with the final instructional segment, no? Yung introns natin, they are processed out or spliced out by the spliceosome mechanism, di ba? The spliceosome what do you call it? Spliceosome facilitated splicing, yan, tinatanggal ng introns and exons are put together. Okay? So these two things, they are independent of one another, but they can occur at the same time, such that you have alternative, I don't know, such that you have achieved expression of or rather regulation of the expressed gene, or in a way modified, no, yung byproduct production. So there are two examples that I'd like to provide. We have the human calcitonin gene for the case of thyroid and neuronal cells. How this gene is regulated is actually varied in these two sets. And then we have sex determination in Drosophila. Let's have an example, the human calcitonin gene. So the calcitonin gene is represented here with a number of exons, as you can see, exons 1 to 5. Okay, and then in between we can have we expect intervening regions, no? Yung mga colored orange. Yeah. So our two systems, our thyroid and neuronal cells, the pre-mRNA can be transcripted with with alternative polyadenylation. So there's poly A tail added, no, to specific portions. Like for the case of the thyroid, there is exclusion of the exon number 5 identified here, and the poly A tail is actually added. For the case of neuronal cells, it's quite different because instead of having unlike thyroid, no, the poly A tail was added to a more a further position or orientation, no. Now, alternative splicing will definitely result to exons being merged together, okay? But because of that, then we expect a difference in the final genetic instruction. Okay, and eventually, translation will lead to the production of the peptide. For the case of thyroid, we have calcitonin, and for the case of neuronal cells, we have our calcitonin gene-related peptide. It's not essentially calcitonin per se in terms of its nature, because it has a different set of some difference when compared to the calcitonin protein. So we call it calcitonin gene-related peptide. Okay, so that's how different genes can be, how varied genes can be regulated. Okay, for the case of inclusion ng splicing and polyadenylation as important mechanisms. For the case of sex determination in Drosophila, we can also expect the same thing. No? In this model animal, sex is determined by the X to A ratio. Okay, so the expression of the sex lethal gene dictates whether the Drosophila will be a male or a female. And that one is actually, in that gene is actually, I don't know, the expression of that is regulated, okay, depending on the X to A ratio. If it, of course, if it's an XX, okay, female, the expression of the gene is without any halt or without any, I know, without any inhibition. So the gene will be transcribed, the pre-mRNA will be spliced in a single manner. While for the case of males, intended males, where the X to A ratio is considered zero point 0.5, no, because it has the Y chromosome instead of another X, the gene will be transcribed. Alternative splicing happens, in but the problem, or not the problem, but what happens is, unlike in female Drosophila, in male Drosophila, the mRNA will not code for a functional protein, for a functional SXL protein. So only the female will translate a functional SXL protein. Okay, and down the line, that protein is responsible for downstream mechanisms or downstream pathway that will eventually contribute to the final sex of the Drosophila. Okay, so yeah, we can see not only at the example of a byproduct can genes be regulated at the level of RNA processing, but also at a very no no a more high level, or a very a more I'd say biologically not superior, but ano na mas a more biologically complex level, which is sex determination, can also be controlled at the level of RNA processing. Okay. All right. So next, let's now move on to another mechanism that eukaryotes can use in order to regulate their gene expression. Let's see. So we have mRNA transport. Yeah. Okay, so before we start with this topic, let's go back, or please remember your notes about nuclear localization. Tandaan nyo yan, no? Balik tayo doon, or kayo mismo, review what you have know about what you have learned from that module. Okay? So what key thing, we know how tight the regulation is when it comes to transporting material inside and outside the nucleus. Okay, so with that, okay, we there we with that concept, we come to terms na at this level, or this mechanism can also be utilized for gene expression regulation. No? There are some studies that have shown that not all transcripts that are produced in the nucleus are exported. No, not almost 50% of them are left in the nucleus and are actually degraded. Only half are proceeding to maturation and can exit to the nuclear pores and then eventually be translated in the cytoplasm. And this process, or yung mechanism na to, where there is the implication of the nuclear pore complex transport mechanisms. Yeah. This mechanism is very tight on its own, and as such, it is really utilizable in when it comes to controlling also how genes can be expressed, or the need for the gene to be, or the genes to be expressed. Okay. Yeah, we have a very nice example here from a very recent paper. The paper can be accessed using this link, so you can check this out if you want to have some further readings. No? So here, I just captured a very important figure from their publication. that gives us an example, or that shows us the RNA trafficking RNA trafficking as a ano no as um, related to, as it relates to sinks kung saan siya mapupunta, and also the mechanisms, no. So here we can see that at the time of transcription, okay, at the time of transcription here, we expect that the growing RNA, that the lengthening RNA, will complex with RNA binding proteins, no. Excuse me. So your elongating RNA that is going to bind to your RNA binding proteins, forming what we call RNA protein particles or complexes. No? So these RNA protein particles or complexes, they are ready for exportation. So alalahanin nyo yung ano natin, yung export and import cycles natin. So what are those proteins that are binding to the RNA for it to be exported outside the nucleus or for it to be imported inside? Um, Once that it is exported out, no, in the cytoplasm, what makes this RNA protein particles or RNA I don't know protein complexes, what makes them transition to certain locations in the cell, for the case of a nucleus, are the I don't know the cytoskeletal proteins, the motor proteins, no? So the motor proteins in the cytoplasm cause the RNPs, the complexes, to either go to one portion, the axon, or another portion of the neuron, yung dendrite, diba? And uh, what do you call this? Eventually, now once that they are in their final destinations, and they may stay silent for a while, they will not undergo translation, or they can once that a signal specific to the activation of that RNA, of that mRNA is present, then there's a time na that mRNA will be translated. Okay, like for some cases, those that are found on ano no dendrites with connections to what do you call this ends of neurons that release neurotransmitters. Yeah, so neurotransmitters can serve as a signal that will bind to receptors bound to the membrane ng dendrite portion, and once they're bound, yan, they can serve a signal to actually activate the RNA to be to be triggered na, okay, it's now time for the RNA to be translated. Okay? Excuse me. So we have our translating ribosomes that are found in found in the area that are proximal to receptors that are specific to neurotransmitters. Okay. Also, ano naman for the case of those staying in the axon, no, the translation might be later, at a later time. Okay, kasi the ones that are nearer doon sa postsynaptic area, or the synaptic area, will of course be given the trigger earliest, no, compared to those that are farther from it. Okay, so at the level of the level of mRNA transport, this is a very good example how transportation of the mRNA molecule, its localization, can serve as a regulatory step, no, or as a tightening step, no. Right. Now let's move on to our third, no, or our fourth rather, um, level or mechanism that genes of eukaryotic systems can be regulated. The expression can be regulated. So we have translation, mRNA translation. Very good example of this is a developing animal. So I think I've already given a few discussions about this before, no? Yung unfertilized egg, no? That as an example, we expect that in an unfertilized egg, the mRNAs that are stored, the pre-stored mRNAs are maternal, okay? And they stay silent until a signal is given or until a signal is present. Primarily, yung fertilization as a signal. Okay. So, at the level of translation, so yung before ano na to, before natin further discuss tong concept, no. At the level of translation, yan, which you can relate to processing na rin in a way. Okay, you have your protective structures, your 5 cap, your 5' cap, and your poly A tail. So these two things are important in the sense that those mRNA that are staying silent for a while or that need to stay silent until they have the trigger or they have the signal. Yan. So these two elements must be present in order to prevent the degradation, no, nung ating mRNA. Actually, the concept is more, I would give more emphasis on this in the ano na, on the fifth mechanism. But essentially, for the concept of mRNA translation, as an example, yun nga, meron tayong ang context na very popular, which I've already discussed, I think, for the second time, is yung unfertilized egg. So your unfertilized egg will have to wait for the signal of fertilization for the translation to be more active, to have higher rates. No? Protein synthesis will be primarily dependent on or will be primarily active for the case of unfertilized egg, mga maternal mRNA, no? Excuse me. Maternal mRNA ang very actively translated. And once that fertilization takes place at a certain juncture, we expect that the fetal made or the zygote made mRNAs will take over. So they're the ones that will sustain the process. No? So yung translation, factor natin yung time. So we factor time and we also have to factor signal. So at the level of mRNA translation, and genes can also be regulated. Okay? Now, the fifth mechanism is on degradation. So, how mRNA decay rates can affect gene expression. We have a very good, example already. Yung kanina, we talked about the protective elements. No? We talked about these protective elements, how they can prevent mRNA to be degraded. So in terms of the RNA products, the most degradable or yung pinaka mataas yung mabilis yung turnover are the mRNA. Your tRNA and your rRNA, usually they're very stable, no? They are long-lived compared to your mRNAs. And again, thanks to the protective elements such as the cap and the tail, the metal cap and the tail, our mRNA is protected, no, from 5' exonucleases and 3' exonucleases that can, degrade the molecule at both ends. If the cap and the tail are absent, no, these two exonucleases can partake in the process of degradation. Okay. Ano pa? Okay. Okay, so this one is a very basic illustration, no? A very basic idea. So the important, what do you call this? The important protective elements. And we have to remember those important protective elements. Now, I have another illustration, another illustrative example for us to really fully appreciate yung how genes can be regulated at the level of translation. let's have this example. Yeah. So this is also a paper. I mean, an example I lifted from a paper from Biomed Central. So this is I think this is a review paper, no? For the case of certain viral pathogens. Yeah. So certain viral pathogens express proteins that target proteins of the host, proteins of the host cell. Okay. So for example, for the case of the herpes simplex virus 1, it expresses the what do you call this? The VHS protein, the viral shut off protein that targets a number of proteins of the animal, of the host cell. No? And its mechanism is through mRNA degradation. So it targets the proteins by degrading the mRNA that code for them. So at the level of, or at the context of pathogen host interaction, yeah, mRNA degradation is actually serving as a mechanism for gene regulation. Kasi imagine these target proteins that they are, of course, turned off or shut off, 'di ba? By the viral protein, then they will not be able to if their mRNA is degraded, their mRNAs are degraded, then they won't be produced. So that's regulating the quantity of these target proteins. So the same case for other pathogens. They express specific proteins that can also target specific that target proteins of the host and different mechanisms of turning them down or turning them off or knocking them off. No? So for the case of the viral shut off protein, yeah, for the case of HSV, yeah, mRNA degradation is the mechanism that is utilized by the pathogen, by the protein, in order to target host proteins. Okay? Alright, so I think we're now nearing to sa ating last few slides. Last three slides. Ayan. Okay. So, uh, for this example or for this mechanism, I want you to go back to our module on endomembrane system. Yeah. So balik kayo dun sa discussion natin on how eukaryotic cells traffic molecules within the cell itself, intracellularly, and um, exportation din. No? So, yan. Maalala natin yung mga concepts such as ER-associated degradation, the unfolded protein response. Yan. Maalala niyo yun. I will leave the review to you. Okay, so let's now finish this lecture by discussing the last mechanism. So we know that not all proteins are expected to to survive for a long time. Okay, so there are long-lived and there are short-lived proteins. And if you look at the central dogma, this is the last stage at which the eukaryotic cell can take control or can influence some level of regulation to how genes will be expressed. No? So there are for the case of overproduce, sabi natin, no? There is a case of an overproduction of a specific protein, and that protein is not really needed by the cell. Uh, what can we expect as the cell's natural response? No? So of course, for that case, yan, we have the actions of our degradatory proteins, mga degradatory enzymes natin. We have proteases, which are essentially enzymes that break down peptides, break down proteins. They can be found normally in our lysosomal vesicles and also the proteasomes. No? Now, if a protein is tagged by a signal by ubiquitin, usually, okay, it will enter the proteasome, so it can be degraded. So there is, you remember protein tagging, diba? It's an important concept when it comes to the quality control of proteins that are found or produced by the cell. But for proteins that are not properly folded, they have to be degraded. Yeah. So one example is proteasome-led degradation. So once that these proteins that are either overly produced or are unfolded, once that these proteins are identified na they should be degraded, then their quantities will decrease because of being tagged as for degradation. Okay? Yeah, so in summary, we have this list of key concepts. So I won't go over them anymore. Huh? So but we just have to I know we just have to go back to the idea that yun nga, in living systems, gene regulation can be expressed in multiple ways, multiple mechanisms, and the elements that are the ultimate elements or the identifiable elements that play critical roles in employing these mechanisms are actually genetic in nature. So yung mga ano natin, genes found in the operon for the case of prokaryotes, and those that are the genes that can also be found in eukaryotes for the case of the structural gene set, and also the mechanisms such as post-transcriptional modification, post-translational modification. And aside from the genetic elements, we also have our, I don't know, protein elements that can participate in the whole process of gene expression regulation, especially for the case of degradation, mRNA degradation and protein degradation. We have enzymes and degradatory groups of proteins that at the final juncture of information flow, okay, they can contribute to the regulation of specific genes and the products that they code for. Okay, so that ends our last module our module on gene regulation expression. Now, a few reminders. You need to complete the asynchronous module on signal transduction within the week. We will I'll be able to upload it before hump day, before Wednesday, and then next week will be for cancer cells. I am still I'm still designing how most effective can we deliver, how effectively can we deliver the last topic, because as promised, or as I have described to you during our course orientation last year, the last module on on cancer cells is sort of a ano synthesis module, because the context of cancer cells, we're going to cover everything, no, from your first module until signal transduction. You will be able to appreciate how each of those modules can be found, how the learnings that you have from them can be reflected dun sa pag-aaral ninyo ng last module natin. So I'm still thinking how I can effectively deliver that to you. So it's either I will record a lecture or I will have interactive content. Okay, but again, that's a problem for next week, not for that's another learning, no, no, learning, I don't know, module, not for this week naman. Okay, so that's it. I think one final reminder. So your enabling assessment, okay? I know that the enabling assessment for this module is really one of the toughest ones I've provided so far. But I would be happy to see, or I'd be happy to check papers that will really show me correct answers. Okay, the paper, as I mentioned, is very hard to read, but I'm pretty sure you had a lot of time, or you were able to utilize your time in reading it. Okay, so I hope that you guys will really be able to deliver very good enabling assessments for this module. Okay, that's it. Thank you.